Studies, a powerful research tool, organized by Pharmacology Department of Bharati Vidyapeeth College of Pharmacy, Navi Mumbai. To brief all about our organization, Bharati Vidyapeeth was established in 1964 by the eminent educationist and visionary Honorable Dr. Patangrao Kadam sir, with the objective of bringing about intellectual awakening. Today, it is one of the large educational organizations in the country with more than 180 educational institutions affiliated to various renowned universities. Bharati Vidyapeeth College of Pharmacy, since its inception in 1993 till today, has achieved many milestones, such as college has established itself as one of the colleges of choice in University of Mumbai, with the untiring efforts of visionary management, Principal Sir, Dr. Vilasrao Kadam Sir, faculty, students, and supporting staff of organization. Our organization had made its impact all fronts. We are proud to mention that our organization has approval of all regulatory agencies, namely DTE, AICTE, and PCI, and is recognized as a research institute under UGC Act 1292F. Our college is the first amongst the private institutes in Mumbai to receive prestigious NBA accreditation. Our college is consistently ranked amongst first 100 pharmacy institutions in NIRF ranking and has permanent affiliation to University of Mumbai. Our organization has emerged as a center of excellence for academic research and is now globally recognized as a pioneer college imparting quality education. We at Bharati Vidyapeeth College of Pharmacy believe in timely updating our knowledge, for which every year our college organizes webinar on current research topics. In today's scenario, the cell culture technology has become indispensable in the field of life science research and also requires specific technical skills. With the same objective, we have organized this national webinar wherein our eminent speakers will be elaborating on the principle and concept of culture techniques as well as its application. I truly believe that the blend of domain knowledge of our resource persons and engagement of participants attending today would certainly make this webinar fruitful and productive. Once again, I welcome you all and wish happy learning. Thank you. Over to Sonam Tanwar to begin with the scientific session. Thank you.
Our first resource person for today is Dr. Abhay M. Harsulkar sir. Sir has completed his PhD in the Department of Botany from Dr. B.A.M. University, Aurangabad. Sir has achieved national scholarship for international students, researchers, and academic staff for the academic year 2019-20. Archimedes Foundation from Estonia. Academic and Diamond Jubilee Distinguished Fellow in Pharmaceutical Sciences, Themis Chemicals from Institute of Chemical Technology, Mumbai in the year 2013. Sir has honored with many prestigious awards such as Excellence in Research Award, Special Achievement Award, Bharat Shiksha Ratna Award, Researcher of the Year Award, and many more. Sir has chosen for career program under the McKnight Foundation Project, Washington State University in the year 1990. Sir had a membership of scientific panel and Fortification, Food Safety and Standards Authority of India under the Ministry of Health and Family Welfare, Government of India from 3rd of Feb 2017 to the 2nd of Feb 2020. Sir is a member of Task Force on Bioenergy and Technical Screening Committee SBIRI DBT from Department of Biotechnology and Ministry of Science and Technology and Project Screening Panel SERB DST Research Advisory Committee and many more. Sir also has membership of Biosafety Committee of Neerat Biosolutions Private Limited and Advisory Committee of We Innovate Biosolutions and Top India CRO in Pune. Sir has done around 11 funded projects, 12 patents. He has published in various renowned journals with impact factor of 611. Sir has published 100 plus DNA sequences submitted in public domain. Currently, sir is a professor and head of pharmaceutical biotechnology department from Pune College of Pharmacy, Pune. With this brief introduction, I request Dr. Abhay Harsulkar sir to please proceed with the session. We welcome you, sir.
Thank you. Thank you for inviting me, and I express my deep sense of gratitude towards the organizers, right, for giving me this opportunity to be here with you all on this platform and share my research experiences with you, right? Okay. Um, I hope I can begin with my talk straight away, and then am I allowed to share my contents?
Yes, sir.
Yes, sir.
Could you see my screen?
Not yet, sir.
I think my share is still inactive, so you should allow me to.
Okay, okay, sir. Just a minute.
Okay, now it's coming.
Yes,
Okay, can you see that now?
yes,
Yes,
it's
sir. Now it's
visible.
visible.
Can you see my screen now? Okay, right. So let's
Yes,
begin, right, with
yes,
cell,
please, sir.
cell line and applications, right? So we basically has divided our talk between Shama, Afre, and me, so that will be serving you something different today, right? So okay, so let me begin with that, right? So my topic entitled is like kind of cell line and applications. So we'll see something what that. Let me begin with a story, like story of Chris Bonson, right? You could see his picture here, and he's Chris Bonson, and he's not more than, like, I'm sure, just a 21-year-old uh, in 1960, and then must be of some same age of many of our viewers, like especially young student viewers, like they are like around 21, and then you can understand. So he was like driving his car, right? And then one morning in March 6th, right? And then he hit upon, and then it was like quite a bit rainy and things like that. And then a skiddy atmosphere, kind of like March is actually a summer actually, but then there was a shower, right? And then there was not a good driving conditions. Somehow he lost his control over his car, right? And then he hit upon a tree, right? And then bounced back and then slammed into a telephone pole, right? That was a fatal, almost a near fatal accident, I should say, right? And then he suffered a serious injuries in his neck, like a cervical spine cord injury that has been diagnosed, and that paralyzed him from neck down. Right? You know that there could be like many such accidents that can happen, and then while you hit upon in a sitting position, because you drive a car in a sitting position, the most vulnerable part can be your neck or your lumbar region spine, right? So you there are two most likely you can get injured. So if you hit upon at a neck region, you end up into a paralysis, right? Why? Because the spine cord carries everything, right? So the spine cord controls our entire body. So if you have a injury, a spine injury in the neck region, right, you get paralyzed from neck side down. So you cannot like move even your limb. And that was the case with the Chris. So he cannot move his arm, his legs, or anything like that, right? So completely paralyzed for almost a year or two. That what happened in 16. So maybe just a 5 year back, right? And then he was taken to a wheelchair almost for a year, right? Lost all hopes, right? That of living, like, and then become a vegetable, right? So he was like almost like doing into that stage, right? But then of course, like, something miraculous thing happened, right? So that he underwent a treatment, right? And of course, like, Chris is not alone. Remember, like, there are something around 17,000 accidents that happen every year, right? So of course, like, it's not like a thing. So he underwent a stem cell therapy, right? And then something called as ASTOPC1 cells has been injected into his spine in his neck region. And then he made a history that he was become the first case, right, of showing a total recovery from that injury. Right. So he is Chris Bonson was regarded as a stem cell man. Right. Why? Because he is the first successful case of stem cell therapy that miraculously cured him, that miraculously relieved him for his conditions. So this is what something happened. Like, you have this you have these neuronal cells. The neuronal cells are covered with myelin sheath. I'm sure you know about that, right? And this myelin sheath is damaged because of this trauma, right? Traumatic injury. Trauma, of course, like, followed by inflammation, and inflammation also is as disastrous as as an injury. Right? So you could see that, okay, the myelin sheath is damaged, and follow here you can see that the conduction is impaired. And because the conduction is impaired through this neuronal cell, what happens is a paraplegia. So either depending on where you hit upon, where the injury is, either neck down region is paralyzed, or the myelin cells, right? And then that has dramatically improved his condition. So this is the first thing that people have heard about a stem cell, right? Because stem cell people, of course, like, people have heard about them, but once you have a case, a patient which is revived, then of course, like, people would look around for that. So these vials, a small vial that contain somewhere around million cells of AST-OPC1, right? Cells which are like oligodendrocytes progenitor cells, right? So they are dendrocytes progenitor cells, and then they become embryonic stem cells, like derived from embryonic stem cells, and when injected into brain and spinal cord, they can turn themselves into nerve cells, right? And therefore functions and communicate with each other. You know, the nerve cells are supposed to touch with each other and communicate with each other, and they started doing that. Then this miraculously this patient is recovered, right? So Chris became the first patient who miraculously recovered for because of these kinds of therapy. Remember, there is no other therapy available, right? You cannot, like, once your spinal cord is injured, you cannot replace that spinal cord, right? There is no spinal cord replacement possible as such, right? But then the stem cell series therapy came at handy for these particular patients. Okay, so this make a big splash all around the world. People started hearing about stem cells and stem cell therapy after that. So not more than just 4 years back or 3 years back, I should say that, right? So these cells even become available today for that. Once we heard this story, then of course, like, we are curious, like, what is a cell, what is a stem cell, and what are the cell lines and things like that, and how we can do with it, right? So let's understand this cell, right? Of course, like you know that we are made up of trillions of cells. Our body is made up of, an adult body of human being is made up of trillions of cells, right? But then this story doesn't start from there, right? Of course, like when we means the life has evolved onto this earth. The life was essentially was a single cellular organism. You could see that, okay, and then a multicellularity arose from that. We are of course a multicellular organism. You could see here one cell as a chlamydomonas cell. So one of the very vivid example of multicellularity comes from the study of algae, right? So you could see that this one single cell is chlamydomonas. A very similar looking cells are here, which are the Volvox colonies. So Volvox colonies shows the first sign of multicellularity, right? And then there is a cell differentiation that happens there, something very important event of cell differentiation. And all that can happen by just by these four processes: adhere, communicate, move, and differentiate, right? So the cells started sticking to each other. People imagine that, okay, if the multicellularity does not have arisen into the life, into the evolution of life, the entire oceans today what we have had would have been turned into just a slimy ocean, right? It is just an ocean of a slime, nothing other than that, right? But luckily enough, right, in the evolution, multicellularity arose. And why multicellularity? Multicellularity has its own advantages, right? So only eukaryotes become multicellular. Remember, right? I'm sure you know about prokaryotes and eukaryote cells, right? Only eukaryote could show that, okay, prokaryote can adhere. They can form a biofilm. I'm sure you must have heard about it, right? But then there is a big problem in communication between the cells. Today we know that in human cells there are lots of different pathways that we talk about, right? We talk about the tyrosine kinase pathway, like the map kinase pathway, and all those kinds of pathways, right? There are many of them are there. There are just few to give an example at, right? But what these pathways do, they communicate, right? So there is a complex pathways, they communicate, and then there is a crosstalk between these pathways, which become the network of communication, network of communicating proteins inside our body, right? And this communication allows the single cell to become multicellular organism, right? So that is what happens here. But in cell culture we are doing the reverse, exactly the reverse. So from single cell we become multi cell. But what we do in cell culture, like I'm sure in the next lecture that you are going to hear, you will see something like that. So you take a mouse, right? You dissect and cut, right? You cut the organ, right? Take this small tissue, chop it into fine part, clearly, add an enzyme into it, right, to digest the connectivity between the cells, make them single cellular, right, and then try to grow them into a media, and then it can go into different passages, and then you can multiply those cells, right? But what you essentially are doing, you are cutting off a cell from a multicellular organism and trying to grow it as a single cell, right? Aren't we doing just the reverse of what a multicellularity been shown by the evolutionary trends? We are, right? We are. What we are doing is isolating a cell from an organism and trying to culture it, right? So that is what we are doing in cell culture, right? So we are just undoing what a multicellularity is, right? Why we are doing that, right? But because with multicellularity, what comes is a cell differentiation. You know that, okay, you are, as I already have told you, that you are made up of trillions of cells. But if you take a skin cell from your own body, you take the neuronal cell, you take a hepatic cell, and then observe under microscope, what you will see? You will see cells which are completely different, right? Morphology, completely different physiology, completely different function, right? The hepatocytes or a hepatic cell from your liver doesn't function as a nerve cells, right? The nerve cell doesn't function as a skin cell. The skin cell doesn't function as a retinal cell. right? So there are like various different functions that the cells are adapted. The DNA from, if you isolate DNA from all of those, I'm sure you know that it's the same DNA. So if I isolate DNA from my chick cells or from my blood cell or from my hair, pluck off my hair and then isolate DNA from my hair root cells, that's the same DNA because it's my genome and that is everywhere the same. But then from the same genome, we arise into a completely different looking cells, right? So if you're, that's what I'm saying, the neuronal cells are different than your hepatic cells, right? And that is what we know as a cell differentiation, right? So we differentiate into a different cells, right? And that what happen into, right, into what we should, when we say about that and cell differentiation, essentially we should talk about stem cells, right? What are stem cells? Stem cells are nothing but the progenitor cells, right? We are the most original cells, right? So we in our body we maintain a population of stem cells. They can replicate themselves, right? So they can maintain a population of stem cell in our body in any given tissue for that matter, and then they also be able to differentiate into various different things, right? So that is what the stem cells are do. And then these stem cells are useful, like as I told you a story of Chris Benson, right? That okay, the Chris has received a therapy from the stem cells. So what really happens with that? That's what we should look into. We differentiate, right, from our zygote, right? So we, from a zygote we differentiate into what is called as a blastocyst, right? And from blastocytes we have, there are primary three layers of tissue that happens: ectoderm, mesoderm, endoderm, and all tissues in our body develop from those. So we essentially has started our life as a single cell, and from that single cell all different types of cells has arise into our body. So that is what I say as this cell differentiation. So we differentiate into the different cells, right? And how the differentiation happens, there's lots of mystery about that, right? We still are understanding the correct sequence of differentiation, right? Correct, the nature of differentiation, we haven't understood that yet, right? But what we have understood is, when we take a cell or take a stem cells and put it in an environment of a neuron, then it develops into a neuronal cells. That is what a Chris has developed. So just 1,000,000 cells being injected into spine cord, and they turn miraculously into a spinal tissue, right? It become a neuronal cell. If you put the cell into your liver, the stem cell into a liver, it will differentiate automatically into a hepatocyte. If you put it into a cartilage, it will become a cartilage, right? And that is why these cells are always in perception with their environment. So there is something that is in the environment that make the cells differentiate into a different types, right? And that is what a cell differentiation means, right? So what will happen, right? When we say stem cells, there are different types. Like stem cell is a cell that have ability to divide indefinite period and give rise to specialized cells. Right? That is what a stem cell is. A progenitor cell, for another, it's like the a little lower category than the stem cell, right? It's partially specialized into a cell that give rise to a particular cell types. I'll give you examples of those down the line, right? But then what is what we know as a stemness of the cell, right? So if you have a cell and it has a perfect stemness, right, that will give rise to a lot of different cell types. But if the stemness is little lower, it's like a progenitor cell, then it will give a typical class of cells, right? And that is how the cells would go and show a different thing, right? For example, this is a bone marrow, and from bone marrow, if you isolate the hematopoietic stem cells, the hematopoietic stem cells change into a multipotent stem cells, a multipotent stem cells into a myeloid progenitor, and from myeloid progenitor you develop into neutrophils, basophils, right, eosinophils, monocytes, right, and then the mast cells, the RBCs, the platelets, and all different types of cells. in your blood, right? Your blood is an assemblage of several different cells, isn't it? Right. So all those cells, immune cells, are there. Where they come from? From a single cell progenitor cell in your bone marrow, right? So that is what we also work into. This is a picture of some recently isolated bone marrow cells in our lab, right? So just maybe 15 days back, right, we received a bone marrow sample. From bone marrow sample, those many cells are being isolated. These are all these are not all stem cells. Of course, it's a mixture of many cells, right? But these are heavily granulated, rounded cells that you could see are these stem cells into it, right? So when you say stem cells, and you also have heard about these words like unipotent, multipotent, pluripotent, and totipotent. So what is a totipotency? Totipotency is this unlimited capability that can give rise to all cell types, right? Into our early embryos. So our embryo, right? Like I told you that we started our life from one single cell. That cell is totipotent, right? The newly formed zygote. The zygote is totipotent because then it gives rise to all the cell types that are present in our body, right? Then there could be a pluripotent, which give rise to the stemness is little less. The pluripotent, where the cells give rise to many same types, like that is like what happened into the bone marrow cells. Then there are a multipotent cells, which can give rise there are different types of cells which gives rise to a few number of different types of cells. And there could be a unipotent cells which can give rise to only one type of cells, like those like cells, like for example in from the testes. So if you take cells from the testes, they would differentiate even into a platelet plate. They would differentiate only in sperms, right? No other type of different cells, right? So they are like unipotent or monopotent. There are different names that has been given, but these are all a totipotency. So what is a totipotency? Totipotency is ability to give all types of cell. I'm sure you have heard about an hydra, and a few cell organism like water में रहता है ना वो, that is a hydra. I'm sure you have seen hydra and studied in your 12th standard. All the cells in hydra are totipotent, right? So technically, if you put hydra into a mixie and blend it, right, and then make a soup and put it onto a plate, every single cell would rise into a new hydra, right? We don't have that capacity, right? You cannot like take one cell from my body and then make me completely out of that, right? That is not possible, right? That is only possible in a different way. We'll speak upon that, right? But then totipotency is therefore limited, right? Especially in case of humans, right? We are like limited totipotency. And this is what happened, as I told you. And most important is the embryonal stem cells. So what the most totipotent cell that we could get for as far as humans are concerned are those cells which are embryonal stem cells. What are embryonal stem cells? This is a fallopian tube, as you could know. That is an ovary, a newly formed hatch egg rolls into a fallopian tube and start moving making its journey down the fallopian tube. Right? It meets with sperms here, become a zygote, get fertilized. The zygote become double cell, right? And then goes into the first cleavage. That's a fusion here. And this is a zygote, right? The first cleavage happens into it. It becomes two cell. Two cell become four cell, eight cell, right? And it goes on multiplying. Finally, it will implant into uterine wall, right? It implants into this uterus, right? And then here you could see a blastocyst, what we call as a blastocyst. So what is a blastocyst? Blastocyst is nothing but like a ball of the cell, a hollow ball, and inside that ball there are more cells in it. And inside those cells, it's called as an inner cell mass, right? And that inner cell mass is the totipotent cells, right? So if we require cells for stem cells for many such kind of purposes, right? So you might have heard about cord cell banking, right, where the cord cells been preserved, right? And then why it is preserved? Why in your adult life, if my cord cell is preserved in my adult life, if I required like any accident, like any paraplegia or any such kind of thing, we can use those cells from my own cord cells, right? But then today we know that we don't need those cord cells. Why? Because we can make our own stem cells. So if I need, I can make stem cells. How we can make stem cells? We'll see that down the line. Okay, but then we can look into, like, of course, like in 98, and embryonal germ cell, EG cells, right, being cultured from gonadal regions, right, of a fetal tissue, right, and obtained from the abortions, right, and that is what is being first produced and published in Proceedings of National Academy of Science, a very prestigious journal. The same year, Science published an embryonal stem cell derived from the inner cell mass of blastocyst, right, and that is what I was talking about, and then shown to be a multipotent or pluripotent cell. So how you derive that? This is the and then you can get it from excess of frozen blastocysts, right? You know that you can do IVF. What is IVF? In vitro fertilization, where you can, in a petri plate, or in a petri plate, you take the egg cell, and then you can take sperm cell, fuse them, right, make them into an embryo, and then keep them frozen, and then implant them into a woman, in her uterus, right? And then it can implant successfully and develops into a baby, which is born by a normal course or by cesarean section. This baby is called as a test tube baby, commonly called as a test tube baby. Why test tube baby? Because the baby is being the it is being fertilized in a test tube, right? Misnomer, not a test tube. It should be called as a petri plate baby, as ideally, but it is like that. Okay, so this is being done. So you take a zygote, goes into a blastula, and then from blastula you could get the inner mass of a cell, and this inner mass of cell you can culture, right, into many different cells, and then you can have those cells for your therapy, right, right, and those cells become available. So there could be, you can use the same technique to clone an individual, right? You can either, if you could take this inner mass of a cell, right, develop into this and then implant into it, it will develop into a baby, right? It's like it become a reproductive cloning, right? So it can become, it is that is what has been done in IVF, in fact, right? So it in assisted pregnancies, you know that you can culture these cells and then implant into back into a mother's uterus. It is born as a complete being, right? We can do that. Like, you might have heard about the same thing being done into animals too, right? I'm sure you have heard of Dolly, right? It's a first shape that has been cloned. How was that cloned? Right, you can clone that by taking a somatic cell, right, and taking an egg. So from egg you remove the nucleus, right, and then take this somatic cell nucleus, put it back into this egg, and now this egg egg gets a trigger to divide. It develops into a blastocyst, and from blastocyst you get inner mass of cell, right? So if you can do that, and from that inner mass of cell, either you can do implant into a mother's uterus, and then you can make a baby, or what you can do is differentiate this inner mass of a cell into various different cell types and use it, right? Either it can goes into blood cells or muscle cells or various different types of cells. So the path of cloning is the same, right? Or path of obtaining stem cells is going to be the same, right? What you could do is just enucleate a egg cell, right, and then renucleate that with a somatic cell, right, and then do it. That is how it has been done. You could see an egg cell there, right, and a needle is inserted into it, and you can remove that nucleus from this, right? And then it's a denucleated. This is the end of a pipette, right, where you can suck. There is a suction there, so this entire cell remains one place. It won't float around. And then you can it's a very microscopic syringe that you can put into it, suck out this nucleus, and then put back another nucleus into it. And then it will go and develop into a blastocysts from the inner mass of a cells. You can take stem cells and then do it. That's what I say. It is not necessary that you need to have a cord cell, right, preserved for you. You can, by using this technique, you can make your own cell and then use it further. Okay, so that is been done, and that you can see that this is an inner mass of a cell, right? It is been removed by another pipette, right, and then utilized for a different thing. So you are harvesting the embryonic stem cells for therapeutic purposes. So this becomes this is another egg, right? It is like being multiple cells being generated, and those cells can be harvested from that and then utilized for the multiple cloning. Of course, there are like issues with it, right? So this cartoon shows an interesting thing that okay, this person say I died, right, for waiting for the stem cell therapy to come up to an age, right, and become available for me, right? But then the stem cell therapy has been perfected till then, and this person eventually died, right? But then this says that I was that embryo, right? So obviously you are like trying many embryos into like that, and that's how the entire thing meets with lots of ethical questions, right? So that is how the things are doing, moving into a different directions, but then still is working, right? Of course, this is a reproductive cloning. I just mentioned about Dolly. She's Dolly, right? And the first ship that is been cloned entirely. So the question is human cloning possible? If you can clone a ship, you can clone a cow, you can clone a rabbit, you can clone a mouse, you can also clone a human being, right? It is not difficult. You can do that, right? You can take this cell. So cell therapy or the stem cell therapy is down the line, but you can use that for cloning purpose. So I can make another me, right? Right, and that is possible, right? Of course, the another me will be a baby, right? It won't be like a big individual like me, right? So it is still a baby. It still has to like take a birth. It has to go to school, learn ABCD, right, and become a scientist down the line, right? So you cannot like clone yourself right readily into a into a into a Xerox copy of me, right? But it is possible to have a clone which is genetically similar to me, not just similar, identical, right? So genetically identical to me, right? Okay, of course, like in farm animals like sheep and cows, we require those for their characters, and then we can use them to clone into it, right? So we can do that, right, into it. But like this apart from cloning, right, how we can do it into a pluripotent stem cells, right? So we can look into a basic uh, research into that, and then how, that is what I told you, that we didn't know, right, how that decision making genes are expressed there. like we have seen and studied some of those genes, transcription factor genes, into the cells that we are trying to differentiate, like stat1, stat6, right, and then even TNF regulatory genes, and then a gene called FOSL1, right? And those genes we have studied, right, in our tissues and in our cells, that what we are working on human cells, and then shown that this transcription factor expression is responsible for the differentiation of the cell, right, from the cell into a differentiation. So if we can know that how to differentiate cells into a different type, right, we can go for a different type of therapies. For example, here, like for example, we take a embryonal stem cell, grew them into an islet cell, and implant them into a pancreas, right? What will happen? Then it will create into islet cells, right? And it will start producing insulin, and then perhaps this patient will will have no need, right, to take injections, insulin injections. So that was thought to be there. It doesn't work like that way. That why it doesn't work that way uh, could take another lecture to speak about that, right? But this was one of the important feature that people started thinking how a stem cell can be utilized, right? So you can, from the stem cell, you can make an islet cell and implant, right? You can develop uh, into islet cell and start producing insulin. Then there could be another possibility of stroke patients. See what happened in the stroke, a part of a brain is dead. And if a part of a brain is dead, right, obviously you cannot revive a dead cell. No, you cannot do that. So what you can do is best is to replace these cells and try to replace those cells. So what you can do is, as I told you, take fibroblasts, they'll induce them into a pluripotency, or take a blastocyst, right? As I told you, from your own cells, you can make a blastocyst, and then these blastocysts are taken, they are mixed together, like with a hope that they will become a neuronal cell and a glial cells, and then you can take the semi-glial cells and put it into the brain. So you can make them convert into a brain cell and then put it into a stroke injured brain, and then this would revive, like the story that I told you about the Chris. This can also become possible, that these cells would become alive. I mean, these are cells are alive, then they would replace these dead cells, and once they replace the dead cell, the the paralysis is recovered, right? So you can there is a hope that the paralysis can be recovered using this type of cell. So what kind of research that has been happening here in case of stem cells? So we are constantly looking the conditions, constantly looking for the factors, right, which can transform this cell into a different type of cells, right, which are useful type of cells. That is what we are trying to do, right? And if you have that portion, if you have, you can make a fluid, you can make a media with all those conditions into it, right? This stem cells become a particular type of cells which can become utilizable for the clinical purpose. So the entire research goes into this direction. So this is what will happen. So take a fibroblast, put it into nucleus, take this blastocyst, take the cells, like, and then you can grow them, differentiate into various different type of cells, may it become neurons, intestinal cell, pancreatic islet cells, blood cells, cardiomyocytes cells. Like, you don't, if you're if the part of the heart is dead, it would be able to replace cardiac cells with that, and then it will become, the heart will become functional. Myocytes can become chondrocytes or skeletal muscles, and once this cells are dead, you can separate those cells by using a fax machine, right? And those fax separated cells then go back to this a particular patient and do it, right? So you can take cells from the patient cord blood banks, and then you can use those cells and then use these cells with iPSC factor viruses. So you can, if you infect those cells with the virus, now this virus is not one which can cause disease. In fact, the virus which cause disease, all those disease causing genes been removed, right? The useful genes are replaced by that. So this virus become only a vehicle, right, for us to deliver those genes, and then it would deliver this gene into it, like only as a vehicle. So virus doesn't mean disease all the time. Like current vaccine that you are taking today is also a virus, right? So that virus been injected into your cells, and then that virus would transform those gene into your own cells, and you start making spike protein and then get immunized automatically, right? So we use such kind of virus, transform these cells, convert them into, they become a colony. Problem with it is what? It is irreversible differentiation. What is irreversible differentiation? So you cannot, once the cell, it cannot de-differentiate, right? If it can de-differentiate, so this is something what we are trying to do it. we are trying to de-differentiate cell. And if the cell de-differentiate, it becomes stem cell, right? From the somatic cell, it would become a stem cell, right? Or from a stem cell, if you de-differentiate further, it can become the totipotent stem cell. Not gone so far, right? But it is possibility. We are trying to do that. If and then it can be utilized for making multiple types of cells, right? Like unlike plant cells, right? I'm sure you have heard about plant cells also. So you take a plant cell, you go into a garden, pluck up a leaf, right? A small portion of a leaf, bring it to the lab, and then culture it, right? You will see that the cell, that one leaf cell would become a mass of callus. Now, what is a callus? Callus is undifferentiated cell. And where it arises from? It arises from the leaf cells, which are already differentiated. So de-differentiation happened, it become a callus, and from callus you can regenerate the entire plant. You can make root, stem, and then it regenerates into a plant, and then you can plant an acre of field with those kinds of small plantlets. And where they arise from? They arise from one single piece of leaf. That is possible to do in plants, but it is not possible, fortunately or unfortunately, whatever you could say, that in humans. You cannot take a small portion of somebody's body fluid, right, or somebody's cells, maybe a skin tab or something like that, and grow it into an individual. You cannot do that, right? But then, if you could have done that, it would have been a different world altogether, right? We are not trying to do that, of course, right? But what we are trying to do that is skin cell, if you could take and into an earlier pluripotent stem cell, pluripotent stem cells for the to make different type of cells. So from skin cells, you can make suppose muscle. That is something a welcome thing. That is what research people are doing, right? That is what we are doing, and that's where we need chemicals, we need certain kinds of cytokines, we need certain kind of bioactive molecules to treat cells and activate these transcription factor genes, SOX2, right, OCT4. There are different genes which can give stemness to the cells, right? And then you can do that, and if you can do that, that you can cure so many different things, right? So that is where we are going into or heading into things. Like for example, you can take a skin cell, convert into neuronal cells, you can straight away use it like for that purpose, right? So either it can be utilizing or modifying certain gene, or edit the genome and then use it further, or maybe just use the embryonal stem cells, like as I told you, right? You can take a somatic cell from my body, take the nucleus, put it into an egg cell, into a blastocyst, take the inner
I couldn't text or pretty much do anything. I was basically just existing. I wasn't really living my life. I was existing. And now, after the stem cell surgery, I'm able to live my life. If I was there and if I was able to thank them, I would just tell them, thank you for giving my life back. Thank you for allowing me to live my life again.
Thank you, right? That's all from my side today.
Thank you, sir.
Now the session is open for questions. If anyone can
Session.
just unmute and start asking questions, please.
Come on, ask questions. That is more important.
Sir, can you tell what is stemness?
Okay. Good question. As I explained, the stemness is the potency. Right? I told you that potency varies, right? From totipotency, pluripotency, monopotency. So monopotency has a limited stemness, right? The totipotency has the maximum stemness, right? So if we increase stemness, mean you are making the cells, making them able to differentiate in more and more different cell types, right? So that is what a stemness of a stem cell is. Its
So means
ability to
can we say that?
yeah,
Okay, so I was asking that only, that whether it is the ability to differentiate.
yep, it is an ability to differentiate into more and more different cell types. Yes.
Okay, sir.
Hello, sir.
Yes, hello.
Sir, myself Vineet Agnihotri. My question is, sir, can you please elaborate on use of cell line on testing of therapeutic drugs?
Okay. See, a therapeutic, I told you also that okay, if a therapeutic drug is there, suppose there is a chemotherapy agent, right? You know that chemotherapeutic agents are very toxic, right? So where they get tested out, right? So if they could test it on something different, like maybe an rat or maybe an animal, right? Or maybe even a primate for that matter, right? And then when I'm going to get an injection of that, I still have a doubt in my mind that will it not be toxic to me, right? Will it be more toxic to me? In fact, it is going to be toxic. That's how it will kill the cancer cells. But that is what I'm fearing to me. But then, if we can take my own cells, right, and test it, and then say that okay, they are not toxic to you, won't I be more confident, right?
Yes,
Undergoing
sir.
that kind of therapy. So that is what it's been tested onto that, right? Just the toxicity I'm talking about. There could be even you can develop a particular drug using human cells. That's what people are trying to do, right? That okay, you can develop a drug and then test it on human cells, show that they are not toxic, show that they are doing what they are expected to do, and then go ahead with the therapy, right? So we are kind of replacing the test animals, and if you can do that, that is good for both sides. It is good for the animals. We don't use them for our experimental purpose because there is a lot of, like, animal activists doesn't like it, right? Or many humans doesn't like it, right? And then plus, of course, it is good for me that okay, I can be sure that it's been tested on my own cells and found non-toxic. Yeah.
Yes. Thank you.
Any
Sir,
other question?
how replaceable is line studies with the introduction of technology like docking and such?
No, your voice is breaking. I couldn't get really what you are saying.
Hello, sir.
Yes.
Sir, am I audible?
Yeah, you are audible now.
So I wanted to ask how replaceable are actual cell line studies with the introduction of technology like docking?
There are different technologies that would come into it, right? Docking is more of a knowledge based kind of thing, right? So when you dock, you know that what is been happening, right? What are the protein, what are their structure, how the ligand is going to bind, like things like that, right? So that data is useful to make a decision down the line. So you cannot say that okay, only docking is translatable right into it, but docking is one piece, right? And then that will make the entire gambit of knowledge that will help us to go ahead further by the by a therapeutic stem cell therapies, right? I don't know if do I hope I have answered your question, but docking is like just one technology. There are many such technologies, and you need all of them.
Okay, sir. Thank you.
Yeah.
Good morning, sir.
Good morning.
Sir, myself is Ravi Vyas. Can I ask a questions?
Sure.
Sir, how we convert the skin cells like transfer into the neuron cells?
That's what That's what I told you about, right? You take a cell, right? You try to express those genes which would like maybe more of a mitogenic genes into it, right? And if those genes are expressed, then the cell would start dividing, right? And then it will de-differentiate. It would become more potent, right? To differentiate into different type of cells, right? That is a crux of the thing. That's the research is about, right? That how you can do it, right? So the entire research into this is going into that how we differentiate the cells and how we redifferentiate the cells, right? So if you take a skin cell, we de-differentiate into a potent cell, right? And the potent cell is redifferentiated into neuronal cells, right? And if we know those conditions to do that, we can have a technology, right? And that is where the research is going into it.
Okay. Sir, actually, one question is more.
Yeah,
Hello.
sure.
Sir, uh, uh, how to help the stem cells in tissue engineering?
Okay, how you can use stem cell in tissue engineering? Okay, tissue engineering would require a kind of a structure where the cells would grow. Usually, I'm sure the next lecture would cover that. Uh, Shama will speak on that. Like there are basic two different types of cell cultures. One is a suspension, and another is an adherent type. So adherent type cells would require some kind of substrate to stick on to develop, to grow, right? So if you could develop, give them a kind of what is called as a scaffold, right, which is made up of some kinds of biopolymers, right? And this scaffold, if you give, then the cells would start growing onto that, right? And if they grow into it, they would develop into that kind of tissue. So I'll give you an example, right? For example, ear. So suppose many people, they lose maybe an industrial accident or whatever kind of thing, right? So now I need an ear. So what can I do? I can take a scaffold, which would look like as an ear, right? And then put it there and grow cells on top of that. So the cells would grow along with the scaffold, and it would become almost live cells all along the scaffold. It will become an ear like that, right? So if you can make a biopolymer like maybe an artificial cartilage, scaffold out of that, and then grow a living ear, right? Like what we have, right? So that is what is called as a tissue engineering. And this is just one kind of that. There could be other types also. Like I told you, like if you want to make a kidney, right? So it is a tissue engineering. So you have to like, or you can even use 3D printers, right? Use 3D printer, put cells into it, so that printer would print cells onto a scan, make a scaffold. The cells would grow for make the tissue, and then that tissue can be utilized to implant, right? So these are all the things on the horizon. So people are trying to do that, right?
Thank you, sir.
Yeah.
Sarji, good morning, sir. Myself, I'm Jagannath Singh, sir. Sir, can you just highlight on placenta storage bank what nowadays it is in the market?
Not placenta storage. It is a cord blood storage banks
Yes.
usually,
Sorry.
right? So what is being done is as soon as the baby is born, right? There is a long cord, like almost like a 1 hand long cord is there, right? So they clamp that baby right near navel. That clamp is usually there's clamped. A plastic clamp is there. They put the clamp. So once you put the clamp, no blood can flow through that, right? And then they cut this placenta, right, and just discard it, right? But if you don't discard that, and if you allow the placenta to drain off, at least maybe around 150 ml blood comes out of that placenta, right? And all that placental blood is rich with such kinds of cells, right? Which is rich with the stem cells, right? The cord stem cells or cord cells. In normal, they are called. They are pluripotent, right? So if you store them into the bank, like in my adult life, say suppose I met with an accident or I required a therapy, right, where I need cells, right? So instead of looking for a donor, what I could do is I use my own cord cells, right? And cord cells are young cells, right? You remember that. So they are young, they are kept frozen, right? They are not like undergone many divisions, right? So they are young, and then once they come into it, they have a good regeneration possibility, right? So if you heard of regenerative medicine, this kinds of stem cells, right, cord cells or mesenchymal stem cells is the staple thing for them, right? So you can regenerate into it. So if my cartilage is worn out, regenerate that. If because of heart attack my heart portion is dead, right, then regenerate that, right, using these stem cells. If brain cell is dead, right, I may face dementia, use stem cells there, regenerate that tissue, and if that regenerates, it would save my condition, right? So that's how the cord cells are being stored, banked, and utilized, right? But I'm sure 90% or maybe even 100% of the audience here, like, will not have their cord blood stored or banked, right? All what we have to be dependent on is using the adult cells in my body, converting them back into higher into potency, and utilize them back into my own body, right? So that is how the current loop is, and that is possible to do it, right? As I explained you take my cell from my body, somatic cell, take a nucleus, put it into an egg cell. That egg cell would develop into an blastula, and then inner mass of a cell if I take, those are almost totipotent cells, stem cells, utilizable for me, right? So that is how the purpose is.
Thank you, sir.
Yeah, thank you.
Hello, good morning, sir.
Good morning.
Yeah, actually, I have a question regarding can you please elaborate some more points on the cell culture of Parkinson's? Like how to isolate and some stuff, like basic principles.
Basic principle is the same. Parkinson is a disease like where the brain cells are degenerated. It's a neurodegenerative disease. So if you look into that, if you take a scan of a brain of a normal person and a person go into Parkinson's, you will see that there is a significant neurodegeneration that had happened. Right. So now what you could do is if you could get these stem cells, right, and then you take a surgery, open the skull, and put those stem cells there, right, and then hope that these cells would go there and regenerate or differentiate into neuronal cells. And if they become a neuronal cell, right, they would take the lost function of my brain, right? And if they take the lost function of a brain, the person is cured from the Parkinson's, right? Parkinson's, dementia, Alzheimer's, you can use stem cells to regenerate. These are all neurodegenerative diseases, isn't it? So wherever there is a degeneration, you can use stem cell to regenerate that, right? And then if you could do that, that will be that, then you can cure that person, right? So the person who was like showing at least an early signs of Parkinson's would miraculously recover, like the case that I have shown here, right?
Thank you so much, sir.
Sir, do these persons show complete recovery or is the recovery only partial?
Depends, right? It may be partial, it may be complete. It depend on the conditions. They depend on so many different things, right? But you can hope that it will recover at least to a great extent. Sometimes even partial recovery is also good, right? Like that Chris case that I have showed you, right? It's even if in a partial condition, that should be fine because he's completely paralyzed, neck down, can't do anything. But now he can use his hands, arms, fingers, right? So now he can talk. He wasn't be able to talk otherwise, right? So everything is like that can be correlated, and then even partial recovery is fine. He may not go into and participate in the Olympic running race, right? Fine, but doesn't matter, right? If he, as long as he can do his own job and become independent. but it depends on so many different factors.
Hello, sir.
Yeah.
Yes, sir. My question is, can you please elaborate on progenitor cells?
Progenitor cells are the same thing, cells like what I told you about the stem cells, but their stemness is a little different. The bone marrow cell is a progenitor cell. Progenitor cell mean what? They are already committed to become a blood cells, right? And a different type of blood cell. So if you take hematopoietic stem cell, for example, it's a progenitor cell, meaning it can give rise to a different types of cells in your blood. They can become blastocysts, they can become the mast cell, they can become the all blood types, blood cell types, right? But they cannot become the neuronal cell, or they cannot become a cartilage cell, right? So that means what? Their stemness is less than complete totipotent cells, right? So they become progenitor cells. We call them as a progenitor cell. So they can become
Okay,
the.
sir. Thank you.
So when we develop cells in laboratory and before administering them into that patient, how can we determine that some of the cells are tumor forming or not?
There are certain genes that are expressed, certain proteins that are expressed on the cells, right? And then if you negate those cells, you unselect those cells which express a certain protein, right? And then may show a tendency to become a tumor, right? That's what I told you, right? You have to do a fax, sort those cells, right, based on the protein on their surface, and choose only those which are the appropriate and then use for the therapy.
Okay, sir. Thank you.
Fax is flow cytometry. So you have to sort cells, right? And then only the correct population to be used for a therapeutic purpose. Any more?
Good morning, sir. Can you please elaborate on FACS machine?
A fax machine is a what should I say? Is a cap. There is a capillary there. Basically, you make the cells go through a capillary. That's a flow. Well, that's what is called. As soon as the cell come out of a flow cytometry, it is bound with some kind of antibodies, right? And then the antibodies has a particular thing which show either a fluorescence, or it has or a separating agent. So then the cell pass through a magnet, right? A kind of magnet. So they deflect because of a certain antibodies bound to them. Others will not deflect, right? And as it deflects, it is collected into a different tube, right? And that is what is called as a cell sorting. So you then sort the cells and then use a specific population. Flow cytometry, but this is in short what is being done.
Hello.
Yep.
Hello, sir. Does
Yeah,
we have
hello.
a control over these cells?
Control over the cell mean?
If we are implanting in a particular, if the cells are degenerated, if we are implanting a new one, their stem cell, so how we can control their growth? Is there
We
any
cannot
way
control
to
their growth.
track their growth?
No, we cannot control their growth. Once they are there, right, we leave it for the cell to grow and differentiate into the neuronal cells, right? And then of course it is being monitored. If they should develop, if they differentiate in the neuronal cell, you will see improvement in the patient, right? But if they become a tumor cell, then of course you have to like remove the tumor.
So there is risk associated with this stem
There
cell
is,
therapies also.
there is a risk everywhere, right? Even if you drive on your face, there is a risk associated. Otherwise, there is what
Okay,
is an
sir.
option?
Yes, sir. Thank
So cells are replace a stem cell in a their property and they'll become sick. So I want to know
Correct.
that how what makes
Is right their ratios, and then there could be many different factors that could come into it, and then they would make the cell differentiate into it, right? Uh, in response to the environment, the cell differentiate. right? And then will differentiate those cells into that particular type is that is what the entire research is. So if you can like develop a media that okay that you put the cell into it and that the cell would become neuronal cell, or you have another media which where you use that media, the cell would become maybe a skin cell, that would be fantastic. So you have to make that thing, right? You have to understand the biology of the cell. You have to understand what is there which is required for it to differentiate those compounds into exactions. Then the cell would go into that direction. That's like a lot of hit and miss approach there. We don't know like what will definitely do certain kind of thing. But that's science. That's the research there. Basically, that is what we are trying to do.
Thank you, sir.
Yeah.
So why this therapy is so much costly in India?
It isn't costly means what it as you could see now, right from the discussion, the handling cell is a very skillful thing. Like there is you require a lot of skill to handle this, right? You require a lot of research that goes under it, right? Which is important to convert those cells into a destination type, right? And for which you require lots of fine chemicals, maybe recombinant proteins, which we are going to use there, or maybe certain types of monoclonal antibodies that would go into it. So it is all very intensive. So it is costly. Cost is a factor which goes in there. So that's the whole idea. But as the thing would develop, become routine, potentially it will become less and less costly.
Thank you, sir. I request you all to turn on your camera for photo documentation.
Okay.
You all can please turn on your camera.
There are 70 others, 78 people there. I hope you understood my lecture, right? And you liked it.
Yes, sir. It was very informative lecture related to the cell line because in our pharmaceutical sector, this is the area we need to elaborate at this time because we are developing a lot of drug, lot of formulation, but we are limited ourselves to pre-clinical studies, or we can say that animal studies only. But if we can elaborate in the cell line, or we can go for the testing, like a cytotoxic testing or studies on cells, so that will be definitely a very great future for us. Thank you for this informative session. I think we should look for many more lectures from you. Like you have said that there is one answer lecture you can give. So I think we expect that lecture from you also.
Yeah.
Thank you very much, sir.
That's fine.
You can turn off your camera. Thank you, sir. Thank you so much for such inspiring story and the inspiring and informative session. The way you enlighten us with basics of stem cells, the cloning of stem cells, the challenges, genetic manipulation, and what are the applications in various diseases. You have made it easy to understand and to apply it in the future research. Thank you so much, sir.
Thank you. It's my pleasure.
We are honored to have you, sir. Thank you. So now, after such a knowledgeable session, we are heading towards our next resource person. Our second resource person is Dr. Shama R. Apde ma'am. Ma'am had done her PhD in biotechnology from the University of Pune with highest grade. Ma'am has more than 15 years of experience encompassing research, teaching, and services in pharma biotech industry and academia with expertise in pharmacogenetic test development and in vitro drug testing. She is expert in optimizing experiments and conducting practicals in animal tissue culture, molecular biology, immunology, protein technology, and pharmacogenomics. Ma'am has published peer-reviewed research papers and a book chapter, SOPs, and laboratory manuals. She has presented scientific research regularly at national and international conferences as well as internal meetings. Ma'am is skilled in laboratory management, including designing, setup, equipment purchase, documentation, as well as recruitment and training of personnel. Currently, ma'am is assistant professor in the Department of Pharmaceutical Biotechnology, College of Pharmacy, Pune. I welcome you, ma'am.
Thank you.
Ma'am, you can proceed with your session.
Okay. I'll share my presentation.
Yeah.
I think it's not showing to share my presentation now. Madam, I think you need to give me permission or something because it's not showing that option here.
Yes, madam, we are working on that.
Okay.
Oh, माझं बंद आहे. Madam, you can try now.
Okay, I'll try. Is it shared? Can you see
Yes,
my
ma'am, it is
presentation?
shared.
Yes, madam, the screen is visible now.
Okay. Good afternoon, everybody. I hope I'm audible.
Yes, ma'am.
Yes, madam.
Okay. Yes. So I'm glad that everybody is so enthusiastic and have participated in question answers and listened to sir. And now we will start how we can do these cell culture techniques and what will be the practical aspects for this. So we will start with a brief definition for animal tissue culture. So, cultivation of tissues and cells from animals outside their body in
Hello.
an artificial environment. And
Hello, ma'am.
yes,
Sorry to interfere,
yes,
but
ma'am.
then your screen is not visible. We can just see your present your screen. Still, the movement is visible. Your slide is not visible.
Uh, okay, I'll keep that. Uh, no, then again, that only option is showing.
But then once you share your screen,
Can
you will
I-
have to minimize Google Meet.
Yeah, okay. Is it now shared? Can you see the presentation?
It is showing that you're sharing. It is not yet visible, but then it is saying that you're presenting. You just wait for a minute.
Oh.
Madam, it is not yet visible.
From here, no, the presentation window option is
Yeah,
not
now we can
showing.
see. Yeah, now we can Google Meet window.
I think you can see my screen, but on my screen, I'm not able to see this presentation actually.
Now, madam, you will have to.
Yes, now it has come. It will get shared. Yes.
Okay.
Okay. Is it visible now?
Yeah, not, but it is showing that. Okay. You just wait for a minute.
Okay.
Yes, now it is visible.
Okay. So slides have shared now.
Okay. Yeah. You. Yeah.
Yeah, is it full screen?
Can
You can
continue,
see?
ma'am.
Yes. Thank you. So
Switch to
now
presentation.
It is not yet screen, madam.
Are you able to see the slide?
Slide is visible,
Yes.
but not as slideshow, madam.
Okay.
Presentation mode में जाओ.
Directly press F5 also, ma'am.
Okay.
I think madam has got disconnected. Yeah.
She's there.
Yes.
Is it shared now?
Yes.
Yeah.
Now go into full screen mode, madam.
Presentation mode. I have presentation mode.
No.
Stop saying. Yes, it is visible now. Yes, it is. Yes, it is visible. Slide is visible now.
Slide is visible, but not in full.
We are trying that. Just a minute. Alright. Uh,
In the tabs, if you can
Madam,
see
what I can,
slideshow again, if
yeah,
file, home, insert.
I
Yeah,
think,
through that slideshow, you can just try once.
yeah.
Click on the slideshow.
Madam, if I send you my presentation, will that be okay?
Yes, yes. Just send it to me.
I'll just send it. Yeah.
Yeah.
Because otherwise
Yes.
everything will look very small.
Okay, okay. I will do.
Yeah. Yes.
Are you emailing it to me, ma'am?
Yes, yes.
Okay.
Same
Okay.
address.
Okay, ma'am.
Yes, it's getting uploaded. Is it now? Type no. Ma'am, I have sent you that mail.
Yes, I will just check, ma'am.
Okay.
I have not received it, madam.
Yes, ma'am.
I have. But have you, if you can try on your device, if you can see the tab file, home, insert, design, likewise, show tab.
Your voice is breaking, ma'am. Just a minute.
Okay.
Is it network issue? It is coming, but coming. Have you received, ma'am?
Even I have not yet received it. There is some network issue even at my line.
Okay, okay.
Just a minute.
It's on my mobile only. Phone से connected है. You need to run.
I'll even put it on WhatsApp, on my personal WhatsApp. You can share it over there. Yeah, I got it now.
ये समझ क्या बोल रहा है? कौन से कनेक्ट करूँ? वहाँ पे भी मैंने हाँ अभी ड्राइव पे मिलेगा. Yes. Okay. Has it come? Screen. Yeah, after that it's coming full screen on my screen, but it's not connecting fine like that in that mode.
So in that Excel, you will have to first close. Probably that is not allowing you to go into slideshow.
Okay. What should be done?
At the start, you can see cell culture. Okay, it has gone. It is showing red in color. Okay, I think it is getting downloaded on my side. Just 2 more
Okay.
minutes. Participants, please have patience. Just cooperate.
वहाँ पे मुझे शो करने के लिए बोल रही है. तो मुझे हो जाता है.
The slide is downloaded and we will start. But madam, I will start presenting it. Present. Madam, you can please.
I'm not able to see the presentation.
Yeah, wait a minute.
Yeah. Desktop पे सेव कर लो.
Is it now?
Should I start with that only?
सांगितलं ना madam
Yes,
you can start from your end because there is some
Okay.
system related issue with Swati madam's laptop.
Okay, I will start then like that only.
Yes. And the content is properly visible. So no worries
Okay,
about that.
okay. Then it's okay.
Yeah.
Again, you will have to let me share. From there, you have to stop sharing. Maybe that option is not coming here now.
Yes, madam.
Yeah.
त्यांना हे द्यावे लागतील ना प्रेझेंटिंगचे. Madam, you can try now. There is no change in our settings from this side.
Okay, I'll check it again.
Yes.
No, that option is not showing, ma'am.
Shama,
Yes, sir.
maybe you can send it to me. I try it from my system.
Okay, sir.
मीटिंग लिंक बताओ
खोला है?
हाँ मीटिंग लिंक बताओ क्लिक करो ना
Come.
Is it visible?
Madam, you can start now.
Thank you. Okay. Sorry for this technical errors, I think.
No, it was from the both sides.
Okay.
Even we are sorry for that.
It's like properly, but today something has gone wrong. Okay.
Yes, yes.
So sorry for the
And
delay.
it's
Now
like,
we'll
madam,
start
even
again.
monsoon weather is
Yeah,
also
actually,
responsible.
yes, yes. Even we are having problem with it's fluctuating like.
Yes, yes, yes.
Yeah.
Okay.
So
Please go ahead,
okay,
madam.
we will start. Huh? Thank you, thank you.
Yeah.
Okay. So with the nice basic understanding cultural cells, now we will actually go to the practical aspects and we will see how we can culture cells, how we can do various cell-based assays, and so all it. Okay. So basically, animal cell culture or animal tissue culture is growing from animal into the in vitro system, which will be the artificial environment will mimic the in vivo conditions. So, and the nutrients, those will be provided to the cells in an artificial means. So, this technique is extensively used in the manufacture and in the testing or drug discovery process. It's nowadays a basic requirement. It starts with in vitro and then it goes to in vivo and then to humans. So even for production of biopharmaceuticals, we need cell culture or a system where we can get the recombinant proteins and even the hybridoma antibodies and all. Everything will be done in the animal cells and tissues. So here we go. So basically, there are three main types in which the animal tissue culture we can divide it into. Like there is explant culture where we culture a small piece of the tissue. Another is organ culture where the entire organ or a small part of it, like a liver culture or small piece of liver you can culture, and that can be used as an organ culture. And if you dissociate the same liver cells and you may make those cells to be in a suspension, the same is called as cell culture. All explant culture is shown. Another one is organ culture. A small piece of tissue is grown, and here the dissociated cells have been grown into a petri dish. So, various requirements are there since these cells are nothing but the living cells, and we are providing all nutrient material, and we have to handle it with very care. And for that, you need to have a good laboratory facilities, aseptic techniques, and then you need to have training in the
Shifa, just
establishment
mute
of primary cell culture, maintenance of cell lines, and cell based assays. All these
Madam, your
aspects we are going
slides
to
are not moving further. You can see the first slide itself.
it's not moving.
No, madam.
What is this happening?
Madam, let me first try whether if I can now it seems there's no net issue. Just let me try it once. Sorry for disturbing.
No, it's okay. Just try once. Is it visible now? Next slide. Am I
No,
audible?
madam.
It's not changing from your.
Let me try once, please.
Okay, okay.
Hello, madam. Hello. Shama, madam.
Yes, ma'am. I'm listening you.
Yes, yes. So we have started from our end.
Okay, okay. Then I'll try. Just a minute, huh? I just try to convert it to PDF and then share.
Yes, madam.
Madam, I suppose my screen is visible to everyone. If you can confirm, I am sharing my screen and it is visible, I suppose.
Yeah, it is visible. Can you check with the changing slides?
So I will, yeah, just a minute. It is on
Yes.
PowerPoint slideshow now. Okay, so I will just mute myself. You can just tell me each time when I have to change slide. Okay? And then again, I will put it in PowerPoint show.
Just okay.
Please start, madam.
Yes, yes. करते हैं start. Okay. So again, we will start with definition of animal cell culture. It's cultivation from the animals into the in vitro conditions where we are cultivating them in a bacteria, where we are simulating in the conditions which are there in vivo. We are providing it in vitro. Can you change? Can you change the slide one? Yeah, thank you. So the basic three types are there: explant culture, organ culture, and cell culture. Where when we come down to single cell suspension culture, it's said to be cell culture. Here you can see here there are explant culture, organ culture, and dissociated cell culture. Next. Uh, So the basic requirements that we will go through, first of all, will be lab requirements. Then we'll go through aseptic techniques, then establishment of primary cell cultures, then how it could be maintained. Next will be the cryopreservation, cell banking, then cell-based assays, and then a bioreactor-based pharmaceutical production, and how it is happening in the pharma industry. We will look into that. Next. So here is the list of basic equipments which we need. Most of the common equipments are there, but specially we will require CO2 incubator, then inverted microscope, refrigerator, and the basically fluorescence microscope, if it is there, that will be good. And if we have the liquid nitrogen storage system, that is very useful for maintaining cells in your stock cultures. So next, ma'am. So basically, when we are providing cells with all the required nutrients and the environment suitable for its growth, we are actually providing the same cell conditions to the bacteria, even if it by chance comes into the cell culture. Here, if you see on the screen, one flask is showing pink in color, and another one is turbid. So it indicates that there is a bacterial contamination. Somehow a bacteria has entered, and instead of our cells growing happily over there, there will be a contamination. So there are bacterial contamination, yeast contamination, fungal contaminations, and mycoplasma contamination that could deteriorate your all cell cultures. Once it comes into one of the flasks, it may spread rapidly, and that is why you have to control each and every point where there is a possibility of getting encountered with any bacterial or fungal, any infection. So you have to prevent at all the steps, and for that you need to take the essential steps to follow the aseptic techniques. Your work area should be sterile. The personnel should be very much trained and keeping hygiene inside the cell culture room. Then your reagents, media, that should be completely sterile. And the technique that you are following and the environment in which you are working, that also should be perfectly sterile condition, so that your cell culture will not be affected by any microbial contamination. So that is why the aseptic techniques are very much essential. Prepare all these equipments, your lab conditions to be aseptic. So first of all, it comes with the preparation of glasswares, plasticwares. So glasswares need to be thoroughly clean, and they should be autoclaved. and the which are n- not sensitive to the heat, those can be done into the say hot air oven. Like glasses without their glass bottle need to take care which atom will go into autoclave, which atom will go to the hot air oven, and the reagents which are heat labile, they cannot be autoclaved. So you need to do media filtration. If you see in this figure, there is media filtration and going on. And for small volume solutions, you need to do syringe filtration. So all these in combination at appropriate places, you need to perform all the sterile operations for your start of any basic steps in the cell culture. So the first of all, the aseptic workbench. After you prepare everything in you have media, glassware, everything in sterile conditions, then you need a aseptic workbench, and that is provided by the biosafety cabinets. So biosafety cabinets are of various levels, like which the basic level where you can handle non-infectious material. Next is the BSL2 facility, where you can handle the atoms which have moderate risk, which have the agents which may cause human diseases of various severity by ingestion or by through touch of that material to your mucous membrane exposure. So basically, we require at least BCL2 facility for cell line work. Okay, and depending on which cells or pathogens, if you want to handle, then you will have to move to the higher level of biosafety cabinets. So once you are done with the BCL2 facility, we will now see how we should arrange all the material. Next step. So here you can see a cell culture lab where laminar flow hood is arranged in a particular way. First of all, you need to clean that with 70% ethanol and switch on UV, make it sterile. Then you arrange your plasticware in such a way that you will not require to cross your hands. And the material which is on the, say, either side, uh, it should be bifurcated. That totally clean area where you can keep your plasticware, your cultures, your media. In the middle of the section, you can work and perform aseptic techniques. And on the right, at the back end, you can keep the things for discard for the biohazard things, where you will keep all the discarded material in that bag. And you need to follow good laboratory practices for all these tissue culture techniques. Next. So now we will go through some of the working instructions, like how practically we are going to do this culturing. So first of all, to eliminate any chances of contamination, whenever we take any bottle or any material inside the biosafety cabinet, we should wipe it with 70% ethanol. Next will be whenever we are opening our bottles or capped, that should be flame. Or when if you are working in a proper biosafety cabinet, then in that case you need to handle that with care, that you will not touch it, because some of the biosafety cabinets will not allow you to use flames. Okay, so opening and closing of your bottles should be very fast, immediate. And next will be the arrangement. As we discussed, it should be segregated, and you can work in between in the laminar flow hood. And for discarding of your cell culture or material, you need to have 1% hypotonic solution, which should be kept ready. And immediately, whenever you want to discard any cells, they will go into hypotonic solution, so that they will not be a cause of any biohazard to anybody. So these are the common practices that we follow for tissue culture or aseptic work technique. Okay, now we will see how primary culture can be done. So here are three basic examples, like mechanical dissociation, Next is enzymatic dissociation, and another one is explant culture. So, first of all, for mechanical dissociation, we need to isolate the tissue. Here you can see spleen is there, and then we have to wash it with PBS and then mince it and chop it with a small light strays. You can mince it and then make a single cell suspension. Some of the tissues are not so soft that they will form a single cell suspension by mere mincing. So you need to add some enzymes, and those will be done with the enzymatic digestion method. So some of the enzymes like trypsin or some other enzymes you can use, which will chew up the small part of your intracellular matrix, and your cells will be released into the media. Then you need to filter that so that the debris is separated from the single cell suspension, and you can get a single cell suspension from your tissue or that organ. Another one is explant culture, where you want to retain the properties of that cells or intact cells you want to regain. In that case, you can go for explant culture, where you take a small piece of the tissue and then you try to culture it. The same will generate into small new cells from the tissue, originating from that tissue, and that gives you the explant culture. Next one. Here is another example of primary culture, where we have done mesenchymal stem cells isolated from the rat femur and rat. Spin it down, and then the host cells are cultured in alpha MEM with 10% FDS. It takes some time for primary cells to attach to the plate, and then slowly they will grow. As compared to the cancer cells or transformed cells, primary cells grow slowly. So they take some time, and after a few days of this is the culture of at least 10 days old when we got these many cells, and after this first passage we moved on to a second passage, and henceforth it is called as a cell line, where we have generated the attached mesenchymal stem cells, all the cells which were born in the first stage itself. So now here we start getting a pure culture of mesenchymal. Next one. Who have passaged it from the primary culture to the cell line stage. Then from that, it is called as cell line. So cell lines can be of two types. So primary culture generally generates finite lifetime cells, and hence they are called as finite cell lines because they have a finite lifespan. Well, some of the cells which are transformed, they can be grown for multiple times, and they can be continuously kept in growing condition as a monolayer or in suspension culture, and that is why they are and drug electricity testing transformed cell. So here are some examples of cell lines, cancer cell lines at various, say at 4x, 10x, 20x. If you observe these cells, you will see the details of your cell culture condition. Your health of your cells can be monitored, and it has to be monitored. This is L929 cell line, which is is fibroblast cells. These are generally used for testing pharmaceutical products or implants. So those which will come in contact with the skin or any organ that needs to be tested with this cell line, and it should be non-toxic to these cells when you want to use any implants for your pharmaceutical or therapeutic purpose. If you see the cells at the bottom, there are the famous cell line HeLa, which is a cervical cancer cell line. Next is MCF7 cell line. If you see the morphological differences in each and every cells, you will come to know they will mimic the original tissue they have come from, or the tumor tissue from which they have originated. So MCF7 cell line is having more of a globular structure. If you see here the next cell line, that is a caco cell line, it is a colon adenomatous carcinoma cell line. So it is showing a difference in structure, and it's if you observe its phenotype is totally different from each other, and their characteristics will obviously be belonging to the respective tissue it is coming from, and that is why they are used as the source as a from human and cells, or that they produce cells from human tissue, and that is why instead of going for animal studies, you can directly see the effect of your drug or test the effect of drug directly from the human cells. So next, here is how it goes into the cell culture. Like whenever you seed that culture, you initiate the cells in the say flask, they will undergo a lag phase. They will initially get adjusted to the environment. They will attach, they will settle down on the bottom of the flask or petri dish, and then they will start growing slowly. So this slope depends upon on the type of cell that you are using. If it's a primary cell, it will grow slowly. If it's a cancerous cell, it will be a steep graph where it will grow exponentially very fast within few days. And in such cases, you need to trypsinize those cells and maintain them or feed them with media at a more regular intervals than the primary cells. If you see here, this phase is the exponential growth phase of your cells, and at this point or at this range, your cells in a very healthy condition. They are young cells which are ready to take up challenges of our any cell culture or testing things. And you need to subculture the confluent. Confluency is the stage where the cells have covered the culture vessel. Friend, when it covers the entire surface of your tissue culture flask or plate. And at this point, the utilization of your nutrients is highest as the number of cells here is high. And then there is also a scarcity of space where they can grow. So it becomes a plateau here onwards. And before this plateau initiates, we need to subculture the cells into different flasks. So depending upon the passage number and the split ratio of your particular cell line, you can split that single flask into 1:10, 1:3, 1:5. So depending on the growth rate of that cell line, you can split them into number of culture flasks. And this is how you maintain that cell line. So before you put these cells into replicate cultures, if you want to test your drug for say by MTT assay, you need to plate your cells in a same amount in each and every well. Okay, so there is a 96 well plate we use for MTT assay, where in each well you will need to add the same number of cells. So for that you need to count the cells. Next slide, ma'am. So this is how you count the cells. This is a Newbert chamber, or it is called as hemocytometer. From one of these sections, you can see there are 4 corners having 16 squares each. So there is a specific pattern in which where you will include that cell and you will count that, and the 4 corners you will write the whatever count you have for after observing this under inverted microscope. So you will count the cells and you take the average of those cells. After that averaging, you will add a dilution factor. You will multiply with it. Generally 1:1 dilution is there. When you take a dye which is needed, that is trypan blue dye is used for viability cell count and cell counting. So 1:1 dilution, say 5 microliters of dye and 5 microliters of your cell suspension if you are you will take the dilation factor as 2, and the factor which has been generated due to the dimensions of this hemocytometer, that factor 1 into 10 raised to 4 is to the your count, and that will give you a per ml count of your cells. Once your count is decided, you can calculate the number of cells that are present in particular amount of your cell suspension. If you want to see few microliters from that, then you need to calculate how much cell density you need for, say, particular 20 ml of culture to get 1 into 10 raised to 4 cells per ml cell. So all these calculations you can do by using this C is equal to N by V ratio. Here you can see a cell counter machine which counts and gives you readily this cell count, which is viable count, dead cells, and the total count it can give in a minute. And nowadays these are also being used regularly. So next one. So further, we will see how these cells will be subcultured to get these cells into suspension. Most of the cells are either adherent to the flask, or they will be in suspension. So the cells which are in suspension, you simply need to spin them down and get them into pellet, and then resuspend it into fresh media. And according to split ratio, you can split them into number of flasks. But for monolayer culture, you need to do trypsinization, and for that you have to follow some steps. First of all, whenever you want to select a flask for subculture, you had to observe it under microscope for its sterility, health of the cells, and the morphology overall, that there is no any cross contamination, there is no bacteria that flask. Then you take it to the biosafety cabinet, and then you do the trypsinization process will be suitable for my assay. If somebody is working on say a rat model, and before start wants to check whether these cells are susceptible or they have their own toxicity on that rat's cells, so he has to select that cells, and the person will select the cell line which is developed for rat. Multiple cell lines which are available for human origin, mice, rabbit, and many more. So you need to select your cell line. So next is the functional characteristics you need to consider. It depends on what activity you want to see. If you want cell lines, if you are using hepatotoxicity, you need to use the cells which are from hepatic origin. So next is whether to go for finite cell line or continuous. Your aim is to get final effect on the primary cells. Then you can use those primary cells. I think presentation has gone. Okay.
Swati madam is trying for the same.
No problem. I thought it is from my side or what?
No.
Okay. Okay, so in that case, you need to check whether those cell lines are suitable for your ultimate aim, like whether you want to go for primary culture, or it also affects whether it's available. It's not possible for all cells to be available at every point. So you have to depend upon the cell lines which are there in the repository in your area. So in that case also, you can go for those infinite cells, which are also actually easy to handle as compared to primary cells. Your cells, if you compare, the finite cells are robust, and they have been acclimatized to the culturing conditions. But primary cultures are not that acclimatized, and they will go for few passages, say 6 to 8, 8 to 10. So it depends on which tissue you are working on. But if you consider the cell lines, they can be grown for multiple freeze them, thaw them, and it makes it available for your experimental use at a regular rate. Are handling it at 37 degrees Celsius very delightfully. They cannot make them or give them shock by transferring those cells information from the cultural conditions to frozen conditions. So over initially you keep it at 4 degrees Celsius, then 20 degrees Celsius for few hours, and again you have to transfer it to -350 freezer. Then again I need to preserve it in a liquid nitrogen. So here is just uh, the content to the slide was like this is a main cryopreservation tank where you can preserve your samples. Now, in this tank, if you want to retrieve your vial, these are the vials where you can keep your cells. Okay, they come in various volumes, like from 0.5 to say 5 ml vials are there. So you need to keep this in a very organized manner so that whenever you want to retrieve any of the vial, you will not disturb all other vials and particularly take out the same vial of your interest. So here are the canes in which you arrange this vial or 4 ml media into that, keep everything ready, and then take out concentration gradient of DMSO is not more than 0.1%. When you suddenly started to switching to the temperature which are at normal temperature or room temperature or 37 degrees Celsius, DMSO may affect badly to the cells. Okay, the one which is preserving at low temperature will affect badly at higher temperature. So you have to take it out. So you spin those cells. Once you have added your cells into media, you have to discard the media content of fresh media, and then you transfer it to the T25. And that flask you can culture her. It also needs to be compact. Okay, now this is a freezing protocol. We have already discussed this. We will go ahead. So coming to the cell, so that cell count should be mentioned. Like, yes, these are cells which are in low amount than usual. Then instead of using say T25, you will revive those cells in a smaller flask. Okay, so while reviving also, you will need this information, and that is why your documentation is very, very essential. Okay. So another thing is the position where you are keeping your vial, because these liquid nitrogen bigger storage capacity freezers are there, which will have so many number of boxes in it, and each row will have say 10 boxes. So having from one vial from that without disturbing others, you need to have very good documentation where that vial is placed in that system. So that is why this cryopreservation records are important. Next. So why do we use these cells, and why do we require so many cells at different time points? And the number of assays or the studies which we can do here are n number of studies. Like you can check intracellular activity. This is very difficult to study it in vivo. Okay, in mice, rat models, if you want to see these minute changes, you are it's very hard to see it with evidence for cell lines to be studied at that level in vivo. In vivo, you have to sacrifice so many animals. The animals have their own variation. Here, if you consider in the cell culture, that variation part is not there. So you get the consistent results. If you put your experiment in say triplicate or in quadruplet, you will have the readings, or that is two quantitative readings with ELISA plate reader, or say different fluorescence or ELISA or so colorimetric assays. You will get replicates which will not have the more deviation, like standard deviation that we calculate in animal studies. Most of the animal studies have more SD, but in cell line you will have the less SD, and that indicates your results are more consistent. That is that's the first benefit of your cell-based assays. And next is you are working at a molecular level. You can work at a cellular level. So intracellular activity like transcription, protein synthesis, energy metabolism, drug metabolism, then gene expression, all these studies, apoptosis you can do, like intracellular activities you can check. You can check the intracellular flux, the proteins which are going in the nucleus, the mRNA that is coming out of your nucleus and working for protein synthesis, that you can monitor. You can isolate the metabolites, you can isolate the receptors on which your drug is working, you can label them, you can stain them. Then the environmental conditions or environmental interactions at different time points, your cells will have different interactions and it will result into different result, isn't it? Your drug, you are applying it to a disease cell line. Then at a disease model, it will have certain different conditions and it will repair. Your drug is supposed to repair it. So what level it has repaired? So all these interactions can be studied. Now cell to cell interactions, even you know how that cells have their own communication system. They have chemotactic communication system. They will release some metabolites, they will release some hormones or some protein. In response to that, the other cell at the other corner of the flask will respond to it and they will find their contact, and then these cells will interact with each other. One of the important properties of cancer cell lines and the normal cell line is like the normal cell lines will have a contact inhibition property. If they come in contact with each other, they will stop dividing, they will stop growing. But if you consider the cancer cells, they will not stop in that way. They have lost the contact inhibition. They will burn upon each other. If you continue to give media and all of the things, they will keep on growing. So all these interactions can be studied. The product, that metabolite products or protein product, all these cells can generate it, and that is what we use for isolating antibodies. Then we use it for development of various peptides, and all these, yes, all these cell products are very essential components of biopharmaceutical industry nowadays. So all these products and these assays are very crucial, and smallest of the say unit, you can measure it in this. And even the genetic analysis you can do for these cell populations, different cells. Though a same person is having a DNA all over his body, but liver cells will express some different genes than the nerve cells, right? So all these things you can actually he monitor or actually he study in a petri dish, and you can grow it with grow those cells, and you can have a definitive experiments on these. Next one. So we will see how cell-based assays are used. So first of all, the most commonly used is cell viability assay, where you can see the how many cells are viable and how many are dead. Next is a cytotoxicity. Those cells which are producing cytotoxic effect, which are really toxic to the cells and they are causing death or they are arresting the growth of those cells, these can be differentiated with these cytotoxicity assays. Then if any product is say making it is boosting the proliferation, okay, so in that case, as in case of Alzheimer's, we need to repair the tissue, right? So in that tissue repair, we need to check how the cells are proliferating, how they are becoming healthy. So cell proliferation assays are used. Now, next level will be the mRNA, protein, receptors, all these targets we need to study. So we label it with different fluorochromes or biochemical, moieties to different antibodies, and that will in turn react with the cells and they will show their signal in case of biochemical signal, or it could be a fluorescent or luminescent signals. Okay, even the cell migration that in cancer cells, it's a very nasty issue of metastasis. Cells do migrate from one organ to another organ. All these migration of these cells we can study using cell migration assay, even cell invasion assay. One tissue, or the cancer cells from one tissue are invading the other tissue. So how this transformation is happening? So how these cells generally we have seen that once the cells are differentiated, they will remain in that stream only. But here you see the cancer cells are going into another organ, and they are developing tumor over there, and it will mimic from some, it will take some of all properties from that organ also. So this is how we have to study, like what is making invasion, and all these studies can be done. Then we have to for biopharmaceutical industry, we are interested in producing various proteins, various peptides, or say hormones or enzymes from these cells. And for that we need to transform the genetically modified system, so that it will enhance the expression of particular or say gene of interest, so that you will get a better product. So all these steps we'll see it in a few slides. Can we go ahead, ma'am? Okay, so first of all, the viability assay. We have earlier discussed like cells stained with the trypan blue, that cells which will take up the dye. Actually, those cells which are dead, they do not have the intact cell wall. So this trypan blue can enter the cell and makes it blue. So by using this, we can test that whether this particular cell is dead or it's alive. If it is alive, it will not take up the dye, and it will be intact, glowy cell will be there. So we will count the number of cells which are viable, number of cells which are dead, and then we calculate the total number of cells and percentage of dead cells or percentage of viable cells. Okay, so this is how we calculate the cell viability. So we can put up the cell growth assays in say 12 well plates, 24 well plates, where we dose our drug on the cells in the replicate cultures, and then we see the effect of that drug on the viability of your cells. Okay, next. Can we proceed now? So next is the MTT assay or SRB, XTT. These are all assays which will show you the biochemical changes which has happened due to the metabolism of the cells. If the cells are alive, they will metabolize this tetrazolium dye, and that will be converted to the violet color or purple color formation crystals. And this property we have used to study the toxicity of drug for the towards that cells. There is another sulforodamine B is another dye which also gives you the same information. So for here, how you can do this MTT assay? MTT assay is commonly required for most of the pharmaceutical drugs. So you need to trypsinate the cells, as we have said earlier, make a cell suspension, single cell suspension, count those cells, then seed those cells in this 96 well plate. Once these cells are acclimatized and have grown in this cell overnight, then you will expose these cells to your different doses of your drug. And along with the standard, you will compare it after say 24 hours of exposure, 72 hours of exposure, or 48 hours of exposure at 3 time points generally. These tests are done, and so that you will get the picture whether your drug is cytotoxic to the cells, or whether it is showing some toxicity after 48 hours, after 72 hours it is showing toxicity. So all these things can be monitored, and this gives you a good quantitative data for your drug, that will indicate whether your drug has any potential for anti-cancer activity. So this is very much popular assay, and used all over the world for testing cytotoxicity. Can we go next, ma'am? So next, as I said, key cell proliferation, and we need to say for regenerative medicine, for stem cells, you can have check your different products which can say generate a metabolic activity, and that is being studied with MTT assay, XTT assay. And not only this, different and other activities can also be monitored. Like this is this MTT and XTT we have just discussed that it will check the cells which are viable, and the cells which will form the purple formation crystal will give you the OD. That control will give you maximum OD, and if there is cell death, your optical density would be reduced. This is how we check MTT or XTT assays by taking its reading as 570 or 590 nanometer wavelength in ELISA plate reader. So next, can we go back? So for our proliferation, we can see the cells by staining. If there is a mixture of cells, then in that case, how will you go about? So if I want to see here that in a blood culture, or how many WBCs have grown, how many RBCs have grown, or what is the different other cells that are being there. So you can label it with different fluorescent dye. Uh, 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 Take a picture on a say fluorescence microscope, and by the using capturing that picture, the softwares are that will give you the counts of those cells. And another is by flow cytometry. As sir has explained, these flow cytometry instruments are very sensitive, and they will be analyzing each and every cell that is going through that capillary. So at least 5,000 cells it requires to stand, and it will take pictures from all the angles for the scatter of light they produce when they come in front of the laser. So these properties can be used, and this data we can use to deduce our information. So for particular fluorescence, a particular antibody is labeled with a particular fluorescence, and that will tell you the amount of that expression of particular receptor on that cell, and the number of cells which are having with a particular receptor will be specific. So accordingly you can calculate, yes, from this mixture these are cells, these are in this much amount, these are in this much amount. So this differential count also you can generate. Next one. So next is ATP concentration assay. You know that whenever you are treating any cells, they will affect its metabolism, and indirectly it will affect the ATP generation and from that cells. And for detecting those cells, you need to lyse those cells. You have to take the luciferase enzyme, which will be labeled, and that you can quantitate by using a microplate reader, and which is a luminescent dye. Now, why do we use luminescent dye instead of MTT? Some of the cells will have a mixture and they may form some debris, but these debris or some of the drugs which are suspension in nature, they will have their own optical density, right? So they will interfere the results. So in such cases, you can use this luminescent assays where this luminescence will be captured and all these other things will be omitted. So depending upon the what actually property you are interested in, we can select from these cell proliferation assays. Okay, next. Okay. Another method by which we can quantitate how much cells have grown is DNA hair synthesis assay. So here, what we are looking into is the amount of DNA that is being synthesized. When new cells are formed, the amount of DNA is also proportionally increasing, right? So we have chosen some molecules like bromodeoxyuridine or ethylene deoxyuridine or thymidine base, which will intercalate the DNA which is being newly synthesized, and that will tell you the amount of DNA that is being synthesized, and that will be the proportionate to the number of cells which are newly say generated. So these will tell you how the proliferation of your cells has occurred. Next one. Okay. Now, as I said earlier, migration. This is the picture where you can see this control cells. We have made a scratch due to which the cells have been removed from that area. Okay, and we will continue this control for the say period of say 7 days, and another set we will treat it with our drug of interest, and that will keep on monitoring. So at the end of your incubation period or the treatment period, you will compare how much that wound that we have created or the scratch we have created has been healed. Okay, so this assay can be seen in two types. Like those drugs which will inhibit the cell migration, they will keep this gap wide open till the end, while those as drugs which have a wound healing capacity, they will heal this drug and they will facilitate the closure of this wound. And so this scratch assay is used for both the things like wound healing assay also and cell migration assay as well. So you can dose dependently calculate this distance, and you can figure out how much inhibition is there for cell migration. Next one. So there is another invasive assay. Here, if you see in this slide, there are filter well inserts. See, these inserts have a filter at the base of these cups, and from where, if you insert it here in the wells, they will have a separate chamber inside. Okay, so at the bottom layer, you will add a medium alone or the test drug which you expect it has some chemotactic activity, and that will may stimulate the cells to migrate down. So in that case you will dose the lower bottom of your well with a chemotactic substance, and you will seed your cells on the top of the filter. So if there is a chemotactic action and your cells are stimulated to migrate, they will cross this barrier as they have the filters, and they will get this chemical signal, and then they will start migrating. If these cells are invading through this well filter, which is actually coated with say extracellular matrix, so they will be mimicking the condition in vivo, and then we are studying this in vitro, so that if the cells we find it in the water chamber, that will indicate that these cells have the invasion capacity, or your product of interest or the test material has the capacity to stimulate this invasion. Okay, so next we can see. Okay, now we have come to the transfection of into animal cells. So what we have to do is we have to maintain and take a good healthy cell culture, then trypsinize it. So there are different methods of this is we have to either surround this DNA with say calcium phosphate, which is positively charged. Your DNA, you know, is negatively charged, and then you coat it with a opposite charged material like a DEAE, and that will facilitate its passage to the cells. Then there is another method where is the negative charge of DNA. So we can use a lipid-based reagent that will form a mycelia, and this mycelia then easily get transformed and they get entry into the cells. Then another technique is directly inserting your DNA by electroporation. Here you give an electric small current shock so that your DNA have cell pores that have been generated by this electroporation. And in this way, you get your DNA transform the cells. Here again, you can immortalize the cell. This technique can be used to insert, say, HPV virions or HPV keep E6, E7 oncoproteins, so that once they are inside, they will keep the cells in a transformed condition and they will continue to grow. Okay. So this is a major application for this. Now this change we can uh, see through and do beta galactase staining of the cells, and we can detect whether the expression is generally the reporter protein, where we can see the fluorescence of cells and we can identify whether so at different stages you can culture, you inoculate this virus, keep it in culture conditions, and then you can harvest those viruses by downstream processing. Can we go ahead? So there is a lot of advantage of using this embryonic techniques. As you can see, with less number of person, you can handle so many eggs, and the recovery and the virus that we can take out from is at a large quantity. So at a large quantity with minimum manpower, you can uh, grow these viruses in embryonic eggs. Now, with the event of advancement in the tissue culture techniques, we have now replaced some of these embryonic egg cultures by the growing them into bioreactors. Different cell lines are grown into bioreactors, and then in those we can grow viruses in a cell culture. Can we go ahead? So viruses in culture or tissues and cell lines, we can grow, and we can identify different cultures for which are suitable for cells that are growing suitably in, say, tissue culture media as a monolayer. They will be anchorage dependent, which are in suspension, which are anchorage independent, and then they will be advantages and disadvantages for various, like suspension cultures, if you say, is easier as compared to monolayer culture. So in that you can modify with using different beads, so then you anchor, and then they produce the more surface area for viruses to adhere to them and then grow, and at the different levels of confluency you can harvest them, and the viruses which you inoculate, they can be checked whether they are giving cytopathic effect. Viruses, can we go ahead? Viruses are majorly cytopathic effect, and even there are fluorescent techniques which can label these viruses or fluorescent antibodies, and that with the fluorescent detection we can check which virus a particular cell line, like here in the first rabies virus or downstream processing. So next, here are the
Okay, ma'am. Thank you for this informative session. It is very elaborated and detailed information you have given to us related to the practical aspect of the cell line. My question is, ma'am, when we do certain immunofluorescence assay, or when we will the interpret, so can you please give some insight on the interpretation of the result when we will do cell viability or cell migration or cell proliferation? That is my one question. Just give us slight insight related to the interpretation of the result. My second
yes.
question is, it is related to that when we develop certain formulations and we do cell viability, cell migration, or cell proliferation studies. So we can see
Right.
is that formulation is toxic or not. But if suppose our formulation is related to the chemotherapeutic agent, so it is a cytotoxic. So
Yes.
in that case, what should be the objective and what should be the result we expect from this cell line studies?
Yes, actually, these cell line studies have mostly developed with major studies for anti-cancer agents only. So those drugs which are really anti-cancer in nature, and they will produce cytotoxicity. Then in that you use the concentrations which are suboptimal to the toxicity concentrations. And in that, at lowest concentration, where it is producing that particular effect, that is affecting, whether it is affecting that molecular pathway that is expected to be getting affected. So those molecular studies, like those studies which will determine the membrane potential differences, or calcium uptake assays, or calcium retention assays are there, which you can study at these concentrations, lower concentrations. Then even if you want to check the apoptosis, whether
Camera on
that
कर मी
cells
screenshot घेते.
are really getting killed. So whether it is through apoptosis, whether it is through other different pathways, that can be studied. And when coming to your earlier question, like at what say, I will take it as like what different effects you want to see those cells in the when the drug is cytotoxic, right? Am I right? Your
Yes,
earlier
ma'am.
question.
Related to the interpretation of the result.
Yes. So mostly when you are using fluorescence based assays, they are particularly targeted to say particular say receptors on the cells, or any particular antibody that is being tagged against that receptor, and that fluorescence will be there only if that particular receptor is there on the cell. If only then and then only it will bind to that cells. If it is binding to that cells, it indicates that that particular cell is positive for that particular say molecule, and then accordingly you can interpret. It depends on what you are targeting. Even the fluorescent dyes can target different polypeptides, which you expect them to get low down, or down regulation is expected. So in that case, luminescence or fluorescence assays are used, because you need their very sensitive assays. And even if there is a small quantity, it should be detected. A biochemical assay is not that much effective in detection of such a small amount. And in that case, such assays are used, like fluorescence or luminescence assays.
Okay, thank you, ma'am. Ma'am, one more question is there. When we will
Yes.
do the confocal microscopy
Mhm.
with this cell line, so can you please tell me how we can generate that slide which are required
Yeah.
for the confocal study?
Yes, actually, for confocal, when mostly confocal is done for those which are growing in three-dimensional structures, right? When we are growing cells in monolayer, we can do the simple fluorescence assay. Okay, fluorescence picture with a simple fluorescence microscopy can be done. But those cells which are growing in the z-axis, like they are growing in three-dimensional, they will have even height also. So in that case, we have to access that Z section, and laser will take at each section, it takes on all images, and all these images are then computer by computer modeling, they will be combined, and they can give you all the structural defined things. And the beauty of this technique is you can target different fluorochromes for different cell structures. And due to that, after time, when you are taking all images of all the sections in a three-dimensional tissue, you can get a more beautiful picture, which is a true picture of each and every organelle structure in the cell. So this confocal microscopy has really turned it into a very nice evidence for all the different structural aspects of your drug discovery. It's drugs
Thank you,
which
ma'am.
are things. Yes.
Thank you, ma'am. I request you all to turn on your camera. We need to take snapshot for photo documentation. Please turn on your camera. And you all can switch off your mic. Thank you, everyone. Thank you so much, ma'am. Thank you for guiding us with all new practical aspects like what are the basic requirements for cell culture laboratory, handling and maintenance of cell culture, selection and cultivation of specific cell lines. You have shared brief information about cell selection and you have shared brief introduction about cell-based assay and an important applications. Then you have also provided knowledge about virus culture and various machineries related with the same. It would be really helpful in our research. Thank you so much. Thank you. The feedback link has been shared in the chat box. You all can fill the form and expected to get your certificate in a week. Now, I would like to request Mrs. Vaibhavi Garge ma'am for the vote of thanks.
Okay, thank you, Sonam. Good afternoon, one and all. On behalf of Pharmacology Department of Bharati Vidyapeeth College of Pharmacy, I am very happy to express our sincere gratitude for the successful completion of webinar entitled Cell Line Studies, a powerful research tool in spite of having small hurdles of internet and system related issues. This actually proves that in the process of acquirement of the knowledge, there is no barrier usually works. So first and foremost, I want to sincerely thank our respected principal sir, Dr. Vilasrao J. Kadam, for his continuous support and motivation for organizing the scientific events. It gives me immense pleasure to express our sincere gratitude for our respected resource persons for today, Dr. Abhay Harsulkar, HOD, Department of Pharmaceutical Biotechnology, Pune College of Pharmacy, Pune, and Dr. Shama Afde Madam, Assistant Professor, Department of Pharmaceutical Biotechnology, Pune College of Pharmacy, Pune, for the smooth conduct of the sessions as well as enlightening us about the very simple and basic concepts, principles, applications of cell line studies, as well as the detailed techniques and equipment related aspects. Our special thanks are due to the webinar coordinator and HOD Pharmacology Department, Swati Hande Madam and Dr. Agarwal Madam for her technical support. The appreciation